Okay, I just want to make a clarification. Last lecture on Monday, I was talking about mitophagy, and there was an error on on this slide. This was previously said part one was meant to say pink one, so that was an error that I made. So pink one is a kinase. It phosphorylates parkin. Parkin is an E3 ubiquitin ligase, and that ubiquitinates outer mitochondrial membrane proteins like mitofusion or VDAC, and then that triggers the steps of mitophagy. So I just wanted to clarify that and correct that. Okay, so now today we're gonna talk about the cell cycle. And okay, so we'll talk about what are the stages of mitosis or cell division. We'll talk about how it's regulated, and that's kind of the major things. There's a lot of pieces involved in this. Some of the recommended reading. Okay, so what is the cell cycle? Why is it important? It's basically important for when a cell divides, it has to give rise to two identical cells, or it has to pass on all the genetic information to the next generation of cells. In order for that to happen, it has to faithfully replicate the DNA and the chromosomes to produce two identical copies. And then those replicated chromosomes and their associated DNA need to be segregated into two genetically identical daughter cells. So in order for this to happen, there's a few things that have to take place. One of the first things is an increase in the cell mass. That's sort of an anabolic metabolism. Remember we talked about the Warburg effect? So that the creation of more lipid and DNA, nucleic acids, and protein. So you need to sort of start to have all the building blocks to make more cells before you actually start replicating DNA. So that coordinating the bulking up with the actual synthesis of DNA and then ultimately dividing the cell into two identical daughter cells. So there's the cell cycle goes through a few processes or stages. So we'll talk in a little bit more detail. There are generally four phases. G1, that's when the cell is usually starting to prepare for cell division. S phase is where DNA synthesis occurs. G2 is after DNA synthesis has occurred and the chromosomes have duplicated, that the cell now starts to prepare for the next stage, which is mitosis. And that's usually where you see the most dramatic morphological changes in cells. So this collectively G1, S, and G2 are referred to as interphase. So this is where the cell generally started kind of growing in size throughout this interphase. It's not necessarily, if you looked at any cell in interphase, it doesn't look particularly unique. It's generally when the cells now start to divide in mitosis, now you see morphological changes. And then one other thing to make note of is that cells can exit the cell cycle and enter into what's called G0. So there's certain times when the cell will decide that it's not the right time to grow and to divide. So the cell can enter into something called quiescence. So it basically steps out of the cell cycle and kind of hunkers down for a while. Now that means if it can re-enter the cell cycle if it gets the proper cues or it's the appropriate environment to do so. So quiescence is sort of a temporary exit into G0. There's another form of G0 which could be post-mitotic cells, so they permanently exit the cell cycle. And then there's one other one, which is senescence. That's when a cell basically, we talked about cells when they reach their Hayflick limit, they're basically permanently locked in G0 and they cannot start the cell cycle again. So, just go over again in a little bit more detail what I talked about, the G1 phase. Again, that's where cells are coordinating the growth and metabolism, which can include the Warburg effect or aerobic glycolysis. And as I said, that's when cells rest when they're not dividing and they enter or exit into the G0 stage. The length of time can vary quite a bit. This is sort of just an average, although this is by no means the definitive amount of time the cell spends. in that phase, but on average, it's about 11 hours per mammalian cells. The next phase is the S, or synthesis phase, and that's where DNA replication occurs, which is typically 6 to 8 hours. After DNA synthesis has happened, the cells move on to the G2 or the F2 phase, and that's where the cells are now preparing to segregate their chromosomes. And finally, mitosis. So this is where we see pretty dramatic effects. The chromatin, we'll talk in a little more detail about chromatin condensation. You'll have the breakdown of the nuclear envelope. Sister chromatids will attach to the mitotic spindle. You get the separation of the chromosomes. Then they decondense and then reform. So what does chromatin condensation mean? It basically means that the DNA and the proteins associated with become more and more compact. So think if you were going to transport a 50-foot garden hose, would you just completely unravel it and drag it along? That's not a very efficient way to move it. You would roll it up and then grab it, so it's a more convenient package to take. So it's kind of similar to what's happening to chromatin, or chromosomes, that because they more tightly associate with the DNA, the histones become more compressed. So that's what's referred to as chromatin condensation. After the chromosomes have separated in the later phases of mitosis, then you decondense the chromatin, relax it, the nuclear membranes will reform, and then finally you get what's called cytokinesis. It's a pinching of the membrane, and then the two cells separate. So cell cycle times vary dramatically. Some cells are able to replicate very quickly. So early frog embryo cells can basically go through the complete cell cycle in less than an hour, 30 minutes. So that's extremely rapid division. If we look at a simple unicellular organism like yeast, and we're going to talk a lot about yeast today, they're anywhere from 1.5 to 3 hours. We already talked about intestinal epithelial cells in the body. They replicate relatively fast, so their cell cycle time is about 12 hours. Typically, most cells that we grow in culture will have a cell division time of anywhere from about 12 to 24 hours. Human liver cells, hepatocytes, divide about once a year. So think twice when you go out on a Saturday night and really booze it up, because you're killing off some hepatocytes, and the other ones that have to come and replace might take a year. And then finally, I talked about cells that permanently exit the cell cycle. Those are what we refer to as post-mitotic cells. The best example of that would be neurons, human nerve cells. So they are terminally differentiated, and they don't divide any further. And those neurons that you have in your brain are generally laid down, for the most part, during embryogenesis, and you have to keep them functioning for as long as you live, which could be 80, 90, maybe even 100 years. So I'll talk just a little bit more about mitosis. It goes through roughly 4 phases. So there's a prophase, it's shown here. So again, we've come through G2 phase, and now the cell's preparing. It's replicated its DNA, replicated the chromosomes. Now those chromosomes have to be segregated. So at the first early phase of mitosis, prophase, is where the nuclear envelope will break down. The spindle apparatus forms. So you have these spindle poles, and these microtubules will attach onto the chromatin. Dr. Damianowski may talk a little bit more about this. As we move into metaphase, the chromosomes align at the metaphase plate. And then finally, this microtubule network, these spindle apparatus, will now start to pull the chromosomes apart during anaphase towards these poles, the opposite end of the cell. And then finally, as we get towards the end of mitosis, telophase, is where the chromosomes, as I talked about, will decondense, and the nuclear membranes will now reform. And then the very last stage is cytokinesis, where there's a contractile ring that forms and squeezes the membranes together. So you probably covered this, I would hope, in high school, maybe in first year biology, so I'm not going to go over it too much. The key thing here, though, is that you want to equally distribute the chromosomes between the two cells. So, you want to make sure that DNA synthesis occurred properly. You completely replicated and accurately replicated the DNA, and then the chromosomes that have been duplicated are segregated equally between the two cell types. if for some reason there's an asymmetric distribution of chromosomes, so one cell gets a little bit more, one gets a little gypped, it's called aneuploidy, and that's generally not a good thing. So that can lead to things like cancer. So you've got, or it can lead to the death of the cell, depending on which chromosomes went where. So I'm hoping Dr. Pemberton is going to get around to releasing this tutorial. It should be happening soon, I hope. I used to cover a big section of the course was microscopy. I talked a lot about phase microscopy, which I know he's going to talk about. So this is a phase contrast image of HeLa cells. Remember we talked about HeLa cells in the first lecture. So phase contrast is a way that we can look at detail inside of the cell. It provides contrast so we can see features. And so you can see here two cells, and you can actually see the nucleus there. There's two cells, you can kind of see the plasma membrane. And one of the cells, this one here, so these cells are in interphase. And at which stage are they in G1S, G2? Kind of hard to tell when you look at them, at least in mammalian cultured cells. Yeast cells are a little different, which we'll talk about in a minute. But this cell now, you can see it starts to round up a little bit. When it rounds up, it forms this, when you're using phase contrast microscopy, it forms this thing called a phase halo. And that's related to some of the optical properties of that instrument. And as the cell becomes more and more round, again, the halo becomes more pronounced. And then finally, you start to see here, there's a little bit of an invagination that becomes more pronounced. And then finally, you get cytokinesis that's roughly occurring about here. You can now start to see the reformation of the nuclear membrane in the two cells, and the cells are now completely separated. So again, most of the cells, when you're looking at cultured cells in the dish, kind of look like this. It's kind of hard to know where they are in the dish, or what stage the cell cycle they are. When you see these rounded up cells with a halo, those are dividing cells, because they're rounding up, and again, you can't really see the nucleus anymore, because the nuclear membrane is dissolved. Okay, so as I said, trying to understand the cell cycle and like maybe using cultured fibroblasts or HeLa cells and just looking at them visually, you don't really see too much unless the cells are undergoing mitosis. And so trying to understand a little bit more about the regulatory mechanisms of control cell cycle, most of those discoveries are actually made in a simple organism, yeast. And there's two types of yeast, Saccharomyces cerevisiae, it's a budding yeast or baker's yeast. And so when these cells divide, they first start to form a little bud. And as they progress throughout the cell cycle, the bud gets larger and larger, until finally the cell splits into two. It has a larger cell, which is called the mother cell, and a daughter cell, which is small. So there's sort of like a little bit of asymmetry in how these cells divide, which is a little unique to budding yeast. But why is that important? Well, you can kind of get a rough idea of how the cell is progressing throughout the cell cycle. When you start to see the very early stage of a bud, that's likely the cell in G1 phase. And cells that have a very pronounced bud would either be in late G2 or perhaps early mitosis. So budding yeast tend to spend more time in G1 phase, so this phase is longer, so you start to see cells either have no bud, or they just have the formation, just the emergence of a small bud. Whereas these steps move a little bit more quickly. And this will be important in a minute. The other yeast type is a fission yeast, Saccharomyces palmae. So as opposed to Cerevisiae, which is more kind of round with a budding thing that goes on, these are like pills, long oval type cells. And the length of these sort of rod, oval-like pill-looking cells becomes longer and longer as they progress throughout the cell cycle. So in this case, they tend to spend a little bit more time in the G2 and M phase. And so that can be somewhat discerned by the length of the cells. So the longer they are, that indicates they're more so in G2, perhaps early phase mitosis. And when they divide, they form this thing called a septum. And they divide equally, so you get two identical cells. So that's a little bit different from Saccharomyces cerevisiae, the budding yeast. So again, elongation occurs this way. They just get longer and longer and longer. Septum forms, and then they split apart 2 equal cells. Budding yeast doesn't work that way. You have a larger cell that's sort of the mother cell, and it starts to form a bud, which will eventually become the daughter cell. The mother cell is always bigger than the daughter cell. And they spend more time in G1. So you'll see more cells with that little bud that's emerging at any particular time, because they tend to spend more time in that phase of the cell cycle. As opposed to these guys, you're going to find longer, rod-like cells simply because they spend more time in G2 and M. Now, that's all great, but how does this tell you anything about the cell cycle? Well, using this simple model organism, it's very amenable to genetic manipulation, or you can expose them to mutagens at a very low level that will cause 1 or 2 hits of mutations within the cell in their DNA. And so by introducing mutations in these cells and seeing how did that affect the cell in terms of its length or its budding capabilities, led to these things called cell division cycle mutants, CDCs. So they created a whole bunch of strains, CDC mutant strains, where they had some changes in their cell cycle. Maybe they were very slow to divide, or they couldn't divide, or in some cases they divided prematurely. And then they were able to look at those genes and figure out, okay, what are the genes that are mutated in this simple model organism? So one of the early ones that was identified, a CDC mutant, was called CDC2. And so when that was, in this case, it was identified initially in Saccharomyces pombe. And so it turned out that the CDC2 is transcribed and is translated throughout the cell cycle. So the cell's constantly making levels of this particular protein. And when they did an analysis of the protein, they found that it encoded for a kinase, the thing that puts phosphate groups on proteins, which I've talked a little bit about already. So this is what they refer to as a cyclin-dependent kinase, or CDK. And so the first one that they identified was in Saccharomyces pombe, CDC2. And then when they looked, hey, is this something similar to this found in Cerevisiae, the budding yeast? And the answer is yes. It was already identified as CDC28. So they are basically the same protein, more or less. There's some slight differences just because they're different species or strains of yeast, but they are both cyclin-dependent kinases. And so if this is, for example, mutated and it's not functional, what happens is the cells try to go through the cell cycle, but they can't. They will bulk up and they will get longer, but they do not actually undergo cell division. So that's where you have a mutation. Under normal circumstances, there's a functional version of that kinase, and the cell can go through the cell cycle quite easily. So when they first looked at this kinase, they started with pombe, they went over to Cerevisiae, and they said, hey, do we find it in other species? And the answer is yes, they find it in every species, every eukaryote, all the way up to humans. And you can actually take a mutant strain, like the CDC2 mutant strain, that doesn't have a functional yeast version, and you can introduce a human version of the CDK, and it can complement, meaning it can rescue that cell cycle deficit. So that, again, when you can take something from a species that's so far evolutionarily removed, and it can still function in a very simple organism, means it's highly conserved. It means Mother Nature, when it does something right, it kind of keeps the plant. It doesn't change the plant too much. That's what we call high conservation function. And so it turns out that many proteins that control cell cycle are highly conserved, all the way from yeast up to humans. So, using yeast as this model organism and kind of making all these mutant strains, and then figuring out what are the genes that are mutated, and then looking at those genes and what are the potential proteins that will code, they came up with sort of 3 classes of proteins that play a major role in cell cycle regulation. I talked about kinases. They put phosphate groups on. Phosphatases actually take the phosphate group off. And then we're going to talk about cyclins. So these 2 are enzymes. This one is not. And because of these fundamental discoveries that were made in yeast, surprisingly, these fellows won, and again, I'm not going to memorize who they are. They won the prize Physiology and Medicine in 2001 for the work that was done in yeast. Again, driving home the idea that basic cell biology and a simple thing like a yeast can provide valuable clues to how we function or if we don't function properly as humans, and how we can get things like diseases like cancer. Oops. Okay, so we've talked about kinases, CDKs. So cyclin-dependent kinase. So the yeast version of this is CDC2, so it is a cyclin-dependent kinase. I know these acronyms kind of get a little bit unwieldy, but I try to keep it down to a low board. So you should understand CDC2, CDC28. We're talking yeast, we're talking cyclin-dependent kinases. Those CDCs, or cyclin-dependent kinases, when we look and a yeast cell, for example, as it's divided and it's going through various stages of the cell cycle, the blue line here represents the amount of protein at those stages of the cell cycle. So it turns out that the cells generally make it and constitutively make it. And so you have a fairly, it doesn't really oscillate much. So the blue line is the amount of the cyclin-dependent kinase protein. Throughout the cell cycle, it does not vary much or at all. The red line is the activity, it's kinase activity. So even though there's lots of this kinase present at G1 and S, the activity peaks at the later stages of G2 or early phases of mitosis. And then it drops off as you progress throughout mitosis. So the question is, how is it that the CDK, the kinase activity, even though it's there, buckets of it, all different stages of the cell cycle, how come the activity only gets turned on at the later stages of G2? In part, it's because of a protein called cyclin. In this case, if we're looking at activation of this kinase for mitosis, it's called, it forms a complex with mitotic cyclin. So the kinase and the cyclin associate together to form what's called the mitosis promoting factor, which makes sense. When those two proteins bind together, that seems to be critical to activate the kinase activity, and to activate it mostly at the later stages of G2 and the very beginning of mitosis. So after it reaches its peak activity, and obviously, what does it do? It promotes mitosis. The name tells you what it does. Once it's initiated the steps of my early stages of mitosis, now we need to shut it off. So its activity drops off. So that's just the first thing to understand. So the cyclin-dependent kinase on its own does not have any enzymatic activity. It has to bind to a mitotic cyclin in order to have activity. That's one thing, but there's a couple of other things that it also needs, which I'll talk about over the next few slides. So this is sort of a cartoon Western blot. It's not a very good one. Excuse my artwork, it sucks. But what this is showing is there's a band there on this blot, called a blot, cartoon blot. So that's the CDC, the cyclin-dependent kinase, and we're looking at various stages of the cell cycle. As I said, the amount of protein doesn't vary throughout those phases of the cell cycle. What does vary is the cyclin. So the levels of the cyclin progressively increase from G1 to S to G2, and then drop off as the cell enters into mitosis. Once the cells divide completely into two independent cells, then the cyclin levels start to accumulate again. And as they progress through G2 into later stages of mitosis, they drop off. That's where they got the name. They cycle, they go up and down, depending on the stage of the cell cycle. So it disappears at one point, and then it reappears at the next stage of the cell cycle. So again, that's what helped, was the first clue as to how do we control the kinase activity. And we're talking about the mitotic CDK, the MPF. So MPF refers to the cyclin-dependent kinase associated with a mitotic cycle. So again, the levels of the CDC2 don't vary. The activity increases, and this is partly correlated, not entirely, with the levels of cyclin. So again, having this complex is really important to activate the kinase activity. But there's this other weird thing I noticed, is that there's actually quite a bit of this formed in terms of mitotic cyclin. It starts to accumulate in G1, and there's quite a bit of it during S phase, and in early G2, there's already quite a bit. So that dashed line refers to the amount of cyclin. So it's certainly low at G1, but at the end of S phase, beginning of G2, there's quite a bit of it. There's quite a bit of this associated. So how come that kinase activity, red is the activity of the kinase, how come it only peaks there when there's quite a bit of cyclin way before that? So there's got to be some other way to activate the kinase appropriately. It's not just the association with the CDC to the cyclin-dependent kinase and the mitotic cycle. Something else is going on. And so we'll talk in a little bit about what are some of those things that also regulate the kinase. So just take a step back and just reiterate the basic concept. In order for the cell to progress through different phases of the cell cycle, the CDCs have to bind to the appropriate cyclin. We'll talk about there's different cyclins at different stages of the cell cycle. So those cyclins, they can increase in certain stages of the cell cycle, progressively increase, and then drop off. And there's more than one type of cyclin. So we talked about mitotic cyclin. That's the one that associates with CDK at the late stages of G2 and early mitosis. But there's other ones. So there's G1 cyclins, G1S cyclins, and S-phase cyclins. And then, as I already talked about, mitotic. So there isn't just one cyclin. There's a bunch of different ones. And they get turned on, their synthesis gets turned on at appropriate times during the cell cycle. And then the protein levels are degraded at later stages when they're no longer needed. So regulation of cyclins, there's two things that regulate it. It has to be transcriptionally turned on. The gene has to be turned on, and it has to be turned on at the right stage of the cell cycle. Once it's accumulated and it's bound to a CDK, and there's some other regulation stuff which we'll talk about in a minute, then the activity, you need to turn off the kinase activity, and that's going to be achieved by degrading that cyclin via the ubiquitin-mediated proteasome system. So this should look familiar because I talked about it last lecture. We talked about E3 ubiquitin ligases. We talked about parkin, that's one, but that's involved in the ubiquitination of mitochondrial proteins. There's many different types of E3 ubiquitin ligases, and there's certain types that are responsible for degrading cyclins at different stages. So just to reiterate, you have a ubiquitin ligase. It'll bind to its target protein. So in this case, the target protein is a cyclin. And then there's also an E2 ubiquitin conjugating enzyme. So this is going to help, this complex of proteins is going to help put ubiquitin onto that cyclin. So to refresh your memory, ubiquitin is a 76 amino acid protein, and there's multiple ones of these ubiquitins that are put onto that cyclin. It's called polyubiquitination. So same principle as what we talked about with parkin, it's just applied to a different protein. So that polyubiquitinated protein is going to be directed towards the proteasome, sort of a big garbage can-like structure, which is appropriate because that's kind of what it does. The target protein is stuffed into the barrel. There's proteases which chew it apart, and the ubiquitin does not enter into the barrel. It's pulled off that target protein and recycled, and it can then be recognized by another ubiquitin ligase, which can facilitate degradation of another cyclin. So, what cyclin is involved in this process? So, there's a number of them that are involved in cell cycle regulation. I'm only going to introduce two. This one is called SCF. So it is a ubiquitin ligase that plays a role in regulating G1 and S phase progression. So it degrades cyclins that are associated with G1 phase and S phase. Okay, so remember, we talked about how we have this mitotic cyclin CDK complex. It accumulates. It actually starts to accumulate even in G1 and S, and then the maximal levels accumulate at the later stages of G2. So, but how come it's not turned on because it's associated with mitotic cycling. In G1, why isn't it starting to initiate things involved in mitosis in G1? Because it needs to be regulated, not just by its association with cycling, but by being phosphorylated itself. So there's a kinase called the V1 kinase, and it puts on a phosphate group on a tyrosine. So why is tyrosine at position 15 in the mitotic cycle? And when it puts that phosphate group on the CDK, that acts as a brake. It says to the kinase, don't start putting phosphate groups in other proteins just yet. Stop the kinase activity during G1 and S phase, and even to some extent G2. So it's important for that phosphate group to be put on when the mitotic cyclin and CDKs associate in G1 and S and early G2. So the V1 kinase will put on that inhibitory phosphate group. And as you get towards the end of G2, now that inhibitory phosphate group needs to be removed. And that will be achieved by a phosphatase. So in this case, it's called CDC25. It removes that inhibitory group, and now when you don't have that phosphate group on, when CDK is associated with mitotic cyclin, it can now be activated and phosphorylate stuff to help promote the movement, the next stages of mitosis. So again, what were some of the ways that we were able to identify this? So if you have a deficiency of V1, so if there's a mutation in which there's no functional V1 kinase, what happens if this is Saccharomyces pontiac is the cells end up dividing very prematurely. So they're very small cells. Why is that? Because there's no kinase, V1 kinase, put on that inhibitory phosphate group. This complex gets prematurely activated, maybe a G2 or S phase. And so the cells now try to initiate mitosis prematurely when they're not supposed to. So they have a very short G2 phase. They have not had the time to properly bulk up and do all the other things to prepare for mitosis. The opposite can happen here is if you have an excess of V1. So if you had a V1 kinase that was turned on all the time, at all phases of the cell cycle, it's going to keep that phosphate group on at all times. And as a result, the cell cannot progress through all the stages of the cell cycle. So it'll get longer and longer, but it cannot initiate mitosis, because you need this complex to trigger all the things that happen during mitosis. So there's two situations where, so V1, where it's not functioning, you get premature activation of the complex, the cells are really small, hence the name V1. It's a Scottish term for small. And if we have excess of V1, so V1 that just gets turned on all the time, the cells are basically cannot progress through mitosis. Now, you can get these phenotypes can also be mimicked by, in this case, if we had a mutation in the phosphatase. So if the phosphatase is not functional, it cannot remove that inhibitory phosphate group. It persists. So now we're getting through into G2, and we cannot activate mitosis. So if we have a mutation in CDC25, and it can't take the inhibitory phosphate group off, that's not going to allow the cells to activate this complex, the mitotic cyclin, the kinase CDK, and so the cells basically are stuck in G2. They kind of bulk up, but they can't go any further. And then again, if we have a phosphatase, CDC25, that is turned on all the time, and it's constantly taking off that phosphate group, you get the same small phenotype. So this complex gets prematurely activated when it shouldn't be, and then the cells will initiate mitosis way too soon. So they have a short G2, and they're very small. So does that make sense? Think about V1, what is it doing? It puts on an inhibitory phosphate group on tyrosine 15. Why does it do that? It's to prevent the premature activation of the kinase during G1S or early G2. But you need to take it off in order for it to be fully active. When is that going to happen? That's going to happen at the late stages of G2. And at that stage, the CDC25 phosphatase is going to take that inhibitory group off, and now you're off to the races. these can be activated. So think about, I love to put questions on tests or exams about what happens if you monkey around with this and what do the cells look like. Hint, hint. Okay. So again, I'm just going to say exactly what I said before, but add a little bit more. So as we're moving through the different phases of the cell cycle, so we're talking about, we're focusing on the MPF, mitosis promoting factor. Say we're doing this in Saccharomyces pond. So they're getting longer and longer. If we look inside the cell, we're going to see an increased amount of mitotic cycling. So we're increasing the synthesis. It starts in, actually, to be honest, a little bit of G1, but okay, this picture says that's it's fine, I don't care. And it's not really important. It's that at this point, we're having the association of the mitotic cyclin and CDK. And as this progresses through G1 and S phase, the W1 kinase is going to become active and put on that inhibitory phosphate group. So it will prevent the premature activation of this complex. Yep, W1, okay. Now, here's another thing, just to add to the complexity. So W1 is an inhibitory kinase. It puts an inhibitory phosphate group at tyrosine 15. There's another amino acid. So kinases can put phosphate groups not just on any protein. They tend to put them on amino acids or any amino acid. So tyrosine is one amino acid. So this is what we call a tyrosine kinase. W1 is a tyrosine kinase. It puts phosphate groups on tyrosine amino acid. Other kinases can put phosphate groups on either serine or threonine. So in this case, this kinase is called CAC, which stands for CDK activating kinase. And it kind of tells you what it does. It puts an activating phosphate group on threonine at position 161. Okay, so at this point, it's almost ready to launch, but we need one last step. We need the phosphatase to come in CDC25 and remove that inhibitory phosphate group at tyrosine 15. And then once that happens, at late G2, this is where we're going to have highest levels of activity. We've got the highest level of cyclin that's accumulated. It's associated with CDK. The W1 kinase, its activity is shut off through some mechanism we don't need to know about. But we're going to activate the CAC. That's going to happen at later stages of G2. It's going to put on that phosphate group, and roughly at the same time, the CDC25 is going to remove that inhibitory phosphate group at tyrosine 15. This is where we have maximal activity, and that's going to occur at the late G2, early M phase. Okay, so what does the MPF do? It's a kinase, and it's going to phosphorylate many things. So some of the things that will phosphorylate are, for example, histones, or other proteins called condensins, which I'm not going to test you on, so don't worry about that. These phosphorylation of these proteins will promote chromatin condensation. So the more compact the DNA is and the histones, they compress, that's condensed. Then we're going to have disassembly of the nuclear envelope. So there's a number of proteins that participate in the structure of the nuclear membrane, which I'm not going to talk about. Dr. Danielowski may talk a little bit about it. And then finally, we have, although we have the formation of microtubules that form the spindle apparatus, we have other microtubules that participate. So we've got microtubules that form the mitotic spindle. Those form. But then we also phosphorylate different microtubules that are involved in the structure of the cell. So usually when a cell is in interphase, it tends to be more flattened. And so we need to, remember we showed for HeLa cells, they round up. So that's because you're phosphorylating cytoskeletal microtubules, which confer the structure of the cells, and you can flatten it out. So the phosphorylation of that promotes the disassembly of the microtubules. And then finally, we don't really want to be focusing on shuttling proteins to the ER, from the ER to the Golgi secreted. We need to shuttle all that down because the energy needs to be devoted towards separating those cells, getting the mitotic apparatus associated, getting rid of the nuclear membrane, and preparing the cell for cell division. So we shut off non-essential stuff. So things involved in vesicular transport will be phosphorylated. and that usually leads to their inactivation. So everything is geared towards mitosis. Other normal cellular functions that are found during interphase, we shut those down. So how is the mitosis promoting factor inactivated at the end of mitosis? Okay, so remember we talked about the cyclins, and we talked about how they get degraded through polyubiquitination. And so how does that happen? Is that mitotic cyclins have this thing called a destruction box. I'm not going to ask you to remember these amino acid sequences, that's just exercise in futility. It's just to know that these destruction boxes of 9 amino acids are one of the things that help promote recognition by ubiquitin ligases. So in the case of the ubiquitin ligase that promotes the destruction of mitotic cyclin, that will occur through something called the anaphase promoting complex, or cyclosome. APC is what I call it. So it's a ubiquitin ligase. It will recognize mitotic cyclins because they have these unique 9 amino acids, referred to as destruction box. I could care less about what these amino acids are. It doesn't matter. But that needs to be there in order to be recognized by the APC. It's a different ubiquitin ligase. So we talked about how during interphase we have increased synthesis of mitotic cyclin. CDK is kind of there all the time, but the activity is basically suppressed. And so as the CDK and the cyclins start to accumulate, we talked about how they get regulated through some phosphorylation steps, and then there's a dephosphorylation of that inhibitory phosphate. 3-161 is there, it's put on, great. So now it's gone through its kinase activation, phosphorylate, all that stuff I just talked about. As we now start to move throughout mitosis, once we sort of hit a late anaphase stage, is that now we need to sort of prepare the cell to now, so now they've maybe segregated their chromosomes. We now need to start to decondense the chromosomes, now to start to reform the nuclear membrane. If the MPF is active, that's completely countered. It's still trying to promote things like break apart nuclear membranes, condense chromatin. We don't want that. As we get towards the later stages and we've segregated those chromosomes, we now need to reform the membranes. We need to decondense the chromosomes. And so we need to shut this off. So that will be achieved by the APC, ubiquitin ligase. It's going to recognize the mitotic cyclin via the destruction box, and it's going to target it for proteolytic degradation via the proteasome. So polyubiquitinates it, and that leads to the destruction of the cyclin. So key thing to note is that the CDK is not destroyed by ubiquitination of the proteasome. It's the cyclins that are. CDKs are generally made, and they probably have a fairly long half-life, and the cell does not go to any great effort to get rid of them. It goes to a lot of effort to get rid of the cyclins. So cyclins are regulated by 2 things. One is their synthesis. It tends to increase at the appropriate stage of the cell cycle. And when it's no longer needed, you need to get rid of it. So the activation of those ligases, in this case the APC, is going to promote the degradation of the mitotic cyclin. And then when it's no longer available, CDK on its own has little to no activity. So this would happen at the very last stage of mitosis, telophase, when the cells have now pulled apart. And again, this is just reiterating what I just said. So again, it's important to recognize all the steps. And so we focus primarily on the NPF. We haven't really talked too much about other cyclins and how they associate with CDK, which I will, not this lecture, but the next lecture. And I'll sort of summarize everything that I just said in this lecture. So we've got CDK pretty well constant. The blue line is the amount of protein, does not vary throughout the cell cycle. The activity gets turned on at the later stages of G2. It's partly regulated by its association with cyclin, although there's quite a bit of cyclin that accumulates it in those earlier stages. That's going to be regulated by phosphorylation. So inhibitory phosphorylation by V1 will happen in these stages. As we get towards the later stage of G2, that inhibitory phosphate group on tyrosine 15 taken on by what? CDC25 phosphatase. CAP is going to put the activating phosphate group on 3D161. That's going to lead to optimal activation of the MPF. And so that in turn will phosphorylate things that promote the destruction of the nuclear membrane, chromosome condensation, all the stuff I just talked about. As we move through mitosis to anaphase, the name gives you an idea. Anaphase promoting complex, APC. What is it? It's a ubiquitin ligase. It's going to recognize the mitotic cyclin. How is it going to recognize the mitotic cyclin? Because it has that destruction box of 9 amino acids that will be recognized by the APC. It's going to promote the polyubiquitination, not of CDK. It promotes the ubiquitination of the mitotic cyclin. It degrades. When does this happen? The later stages of mitosis. When? Anaphase. The name tells you this stuff, right? And so at this stage, the CDK is still intact, the mitotic cyclin is degraded, and the activity of the kinase drops way down towards the later stages of mitosis. And so that's basically the main things to take note of. Okay, so I think just that I don't want to really rush it. I think we'll focus on a little bit more detail about S phase and the other cyclins associated with G1 and S. Now, before we go, I just want to say that you're going to be tested on, this Sunday is the next test. You'll be tested on everything up until this slide. Okay? And we'll have a little bit more detail. So there'll be 20 questions, multiple choice, very similar to last year. Yeah, there's like so many different possibilities. So there's a whole bunch of other steps involved. How do you change just kind of like a very without getting into super detail. So you're right. It's really black and white stones they have the same control.